For a lot of us, animals can be seen as a source of comfort, whether it's pets observing animals pets observing animals in nature or watching a funny animal video on YouTube. Animals can make us feel happy or relaxed. It made me wonder exactly how much of an impact animal presence has on a person. Humans and animals have been interacting for over thousands of years, and humans have benefited from them in many ways. One way more recently people have been benefiting from animals is through animal assisted therapy. ATT is when animals are used for treatment sessions. Most people typically think of as dogs for animal assisted therapy, but in fact, many animals can be used for animal assisted therapy. There has been much research done on the positive impact animals have had on people. In one study in 2013, it showed that older people participating in animal assisted activities had overall higher self esteem than those who did not. In another study done by Hall about disaster victims, those that were rescued by a team with a canine unit were more comforted in the presence of the canine. There's been much research done on how petting an animal can reduce a person's blood pressure and be relaxing. For example, there's a study done by Shiloh where they had participants hold and pet an animal before a stressful activity. In the study, the participants either held a live animal or a stuffed one. They either had a hard shell or fur. The results showed that it did not matter if they had a hard shell or fur, that just holding the live animal had more effect than holding a stuffed animal. This study, however, did not show if the animal had a calming presence or if it was just a distraction from the stressor. The present study was extended to this research by adding a distraction group, an animal watching group, and along with an animal holding group and a group that had no interaction with animals. For my hypothesis, I predict that exposure to animals either through holding, observing will reduce stress more than a distraction or nothing during a stressful situation. For my participants, I recruited by sign-up sheets and asking. I had 22 participants. The age range was between 9 and 35, and the average age was 19. I had 19 Caucasians, 2 Hispanics, and 1 African American. There was 8 males and 14 females. So the design of this study had 4 randomly assigned groups. There was an animal holding group where they held a baby chick. There was an animal watching group where they watched a turtle. There was a word search or the distraction group, and then there was a nothing group where they did nothing. For my procedure, participants came into the room and were exposed to the stressor, which was a tarantula, and she was in her tank. They filled out the informed consent and the demographics, and then I checked their pulse with a finger checker. And then they were exposed to one of the 4 conditions. I then told the participant they may need to hold the spider, but I had to leave the room and get something first. Participants were left alone for 2 minutes with the spider and the condition. I came back into the lab and had the participant take the state trait anxiety inventory for adults, and I also took their pulse one more time. And then eventually I told them they did not actually have to hold the spider. They were debriefed and they could leave the lab. For my pulse results, we used a mixed factorial ANOVA with a pulse 1 and pulse 2 as the repeated measure and animal or control as the in-between subjects factor. There was a significance found between the chick group and the nothing group. What was interesting was that holding the chick actually increased the average heart rate while the nothing group lowered. My thought was that people got so excited to hold a cute baby chick that it made their pulse go higher. So to test this, I had 4 people outside of the experiment hold the baby chicks, and 3 out of the 4 people, their pulse went up as well. For the questionnaire results, we used a one-way ANOVA, which shows the means for the state anxiety. For the 4 conditions was significantly different. The p-value means that the null hypothesis were true. There was only 2.9% of getting this, so the null hypothesis was rejected. However, we did a post Hawk test to compare the means, it was showed that the nothing group was significantly different than the other groups. The other groups were not that much different from each other. So in conclusion, the data would seem to show that the animals were just a distraction from the stressor. So for my discussion, for this experiment, I had a pretty small sample size. So maybe if I had a bigger one and more time, it might have shown a difference between the 3 other groups. I think this experiment could be interesting for the future research if someone wanted to recreate it by trying other animals and other forms of stress to see if there's see what their outcome would be. Thank you for listening. Any questions?
If you could use, like, if you could redo the study, would you switch what kind of animals you could use? Do you think that that would make a difference, or would you keep the same ones?
The chicks, I really like. The turtle, he was actually more psychotic than I thought he would be, so, but he was good to watch. So yeah, my animals were pretty good.
Okay.
Why
Did you,
didn't you have, sorry.
oh, sorry.
Go ahead, Kate.
Did you get a sense if the participants were anxious about holding the spider? Like, was there anyone that was like happy to hold the spider or like thinking they were going to hold the spider?
I did 2 kids. One of the kids was terrified, but the other kid, he was game for holding the spider, so it was like very mixed.
Okay.
Some people were pretty scared and other people were like, okay.
Why did you have them only look at the turtle and not hold the turtle? Because in some sense you got a difference between the chick and turtle are both different in type of animal and their interaction with it. So I was wondering why you didn't have them hold the turtle.
I didn't have them hold the turtle because he was really jumpy and active for a turtle and he kept scratching people.
Okay, makes sense.
Mhm.
Go ahead, Skip.
I was putting myself in this position, and I was wondering, with the chick choice in particular, I was thinking that I get nervous holding small birds because I'm afraid of hurting them, I have the experience of them being delicate, and therefore, it's not the same as petting a dog or even a cat for me. Did you consider that?
Yeah, nobody really seemed to have that. They were really gentle when I kind of showed them how to hold it so that they would be safe with it.
Okay, good.
Also, just as a thing back from the old days of psychophysiology, yes, increased heart rates could be more nervousness, but increased heart rate could also represent increased interest, increased cognitive activity. You see that trend as well, that when people sort of really get involved in it, heart rate also goes up that way as well. So it could be they were just really, like you said, sort of really interested into this, holding that chick.
Yeah.
Was there a reason why you picked the animals you did? I don't know if I missed that or.
Well, originally I was going to have people hold the turtle and watch the turtle, but then, as I've said before, the turtle was a little bit nutty. So then I live on a farm, so I have access to baby chicks, and so then I use baby chicks, which are not usually crazy. So.
How did you decide to use the tarantula as your stress-inducing animal?
The Shiloh study that I was talking about earlier, I kind of based it off that. They used the tarantula, and then Dr. Collins said it would be cool if I could use a tarantula too. So that's where we got it from.
Kudos to Dr. Collins for her office becoming a menagerie to do this study.
Yes, thank you, Dr. Collins.